Hi students, let's talk about statistics. So the discussion you just had about when is a number that's a little bit less than $10 close enough to $10, that's essentially statistics. In other words, the way I like to think about statistics is sort of, are these numbers, quote, different enough to actually matter? So statistics is really, really important for science because in science we want to be unbiased, right? And statistics is one way to help us to do that. So statistics is essentially just a field of math where you're going to analyze differences between groups of data. So for example, you might have an experiment where you have treated some group with a placebo and some other group with a medicine, and you are comparing effects between those two groups. You would need statistics in order to do that. So statistics is essential in science to help us determine if results from experiments are really truly different, or if any little tiny changes we see are just like random chance, just random fluctuations. So in other words, we need stats to tell us, do those differences actually matter? So here is an example to help you. So a lot of times in your math classes, maybe up until now, you will have had to do something like this, where you'll get a whole bunch of data points, and you might have to find the average or the mean of those data. And you have two groups of data, A and B, and you do that and you get these numbers. So the average for group A is 12.7, the average for group B is 14.3. Those are obviously like literally different numbers, but are they different enough to matter? And to answer that question, we need to know something about all of the data points. We need more information than just the average. So one way we could do that is by looking at like a scatter plot here that has literally every single data point in it, and that's what this is. And so if we look at that, I hope what you can recognize is the points for group A almost completely overlap with the points for group B. Like the range is very similar between the two. So those groups actually don't look very different, right? It's almost like all the difference in this average is just because of these few outliers at the top and the bottom. So it looks like these groups are actually not going to be any different, and the little differences we're seeing are just due to random chance. So we would say that statistically, these groups are no different. Even though, yes, the averages are different numbers, but from like a science point of view, as scientists, we would say, no, these groups are just the same, and that little difference we're seeing, that doesn't matter. So in science, that threshold that we use for significance is actually pretty strict. So the term we use for that threshold is called alpha. And in general in science, we use a threshold of alpha equal to 0.05. You can kind of think of that like 5%. We'll talk more about this throughout the year and kind of get a deeper understanding of what that means. But for now, I'm going to really oversimplify it. Please don't tell your math teachers I am explaining it this way, but you can kind of think of it like a percent. So for example, with the example we were just talking about, if you lent your friend $10, sort of the threshold of like what's different enough would be 5% of that, which would be $9.50. So if your friend bought you lunch and your lunch was less than $9.50, that would be the threshold where we would say, no, that's too low. Those numbers are different. Okay, so when that happens, when numbers are different enough to like meet that threshold, we say that those two groups are statistically significantly different. So in other words, we would just say those groups are different enough to matter from a scientific point of view. We think that those differences are really true. So let's look at that data set we just looked at before. So once again, these averages for these two groups are different, but we figured out that because they overlap so much, that they're not actually different enough to matter. So we would say that these two, Group A and Group B, are not statistically significantly different, and any changes we're seeing between the two are just due to random chance. On the other hand, here's a different set of data, so Group C and Group D. Now when we compare these, we can see that, yeah, there are a couple data points that are kind of in the same range, but they're much more different from each other than Group A and Group B were. So these two groups, Group C and Group D, we would say are statistically significantly different. And this represents a real difference, a difference that matters. So there's lots of different ways to test statistical significance, and one of them uses something called error bars. So error bars are just a way to visualize the amount of variation in a group of data. So far I've been showing you these scatter plots where you can see literally every single point, and that's a really nice way to see variation. So here, Group A, those points are like really spread out, so there's a lot of variation in that data. They're just occupying a really wide range. Whereas group C, you see how all the data points are like clustered together? That data set has like almost no variation. So just really, really tight together. It's important to know that variation isn't necessarily a bad thing. It might just be a natural thing, and that's okay. It doesn't mean that the experiment was bad or like the scientist made a mistake. It might just be, that's just how it is. So we can represent that variation. Instead of having to show all these data points, we can do it with error bars. In these examples, the data points aren't so bad because there's only like 15 of them or something, but imagine that there were 100 data points in each set. It would be like, oh my God, so many dots. So that's when error bars are really nice. So this is the exact same data, just represented a different way. Just a bar graph where normally you draw your bar at the average, just like we always do. But now the error bar is telling us something about the variation in that data. So you can see that group A has a big error bar, whereas group C has a little teeny tiny error bar. So there we go. So how do we use error bars as a shortcut to test for statistical significance? It also do with overlap. So let me show you what that means again. So once again, here's our data, and we can show that using error bars like this. So when I say overlap, imagine that, see how there's like it looks like an eye almost, like the top of an eye. Imagine I kept drawing this over. Oh my gosh. Look, if I did that, boom, it literally would point, it would touch that other error bar in group B. That's what I mean by overlap. Like they literally are occupying the same range of numbers, those error bars. So because the error bars for group A and group B overlap, that's like saying, well, the variation between those groups is like occupying the same range of numbers. So we would say those groups are not statistically significantly different because their error bars overlap. On the other hand, if we have two groups of data like group C and group D here, and we do the same thing, we draw that error bar over, and look, it doesn't touch group D's error bar at all. So we would say that these error bars do not overlap. In other words, the groups are statistically significantly different because their error bars do not overlap. So this is a shortcut for us to test for statistical significance. So once again, key point here, this is so important. It absolutely 110% will show up on the AP exam, and you're going to see it on every summative between now and then. So if error bars overlap like they do in these two groups, that means that those groups are not statistically significantly different. In other words, as scientists, we're going to treat those as the same. On the other hand, if the error bars do not overlap, then those groups are different enough to matter, and we would say they are statistically significantly different.